To acknowledge that I want to take a moment to acknowledge that the University of Washington sits on the traditional lands of the Duwamish people, who are now mostly members of the Muckleshoot Nation. Today, it's my great pleasure to introduce Professor Sara Walker from Arizona State University. Sara is the director of one of the new NASA ICAR teams called Planetary Systems Biochemistry. I think she's going to be talking about that a little bit today. Before she arrived at Arizona State University, she began her career at Cape Cod Community College before transferring to finish her bachelor's degree at Florida Tech. She then got her PhD at Dartmouth University, followed by a postdoc at Georgia Tech. And then after that, she accepted her professorship at Arizona State University. So today she's going to be telling us about inferring the laws of life from physics and on a planetary scale. And if you've been in these seminars before, you know that the format is that I'll ask all of you to please turn off your video and mute your microphones. Most of you have already done that, just so that there's no extraneous noise and that Sara can have all the bandwidth she needs. If you do have a question, feel free to type it into the chat during the seminar, or you can hold on to it until the very end, and then you can raise your virtual hand and I will call on you to ask your question to Sara directly. So with that, I'll turn it over to her and let's hear your lecture.
Great. Thank you so much. Thanks for having me come today. And it's been a lot of fun talking with people all morning. So I've had a lot of great conversations already. And I wanted to start today by just kind of giving sort of a broad perspective on thinking about astrobiology as a discipline and where it's going, because I think this really informs how we need to think about the life detection problem and how we should be thinking about the nature of the problem we're trying to answer as astrobiologists. And of course, I know a lot of people, particularly at University of Washington, have been thinking about these things for a long time. And Vicky's been really leading sort of recent efforts thinking about standards in our field and sort of long-term future for how we should be thinking about astrobiology. So I'm setting a sort of 100-year time frame just in the introduction to this talk, just as sort of a thought experiment. And I'm trained in theoretical physics. So when I'm thinking about the problem that we're trying to solve as astrobiologists, as far as trying to discover other examples of life or even understand our own origins, really that breaks down to me for the fundamental problem of understanding what the phenomena of life actually is. And we have a long history in physics of trying to derive deep explanations for the sort of features that we observe in the world and building mathematical theories that allow us to predict interesting features and discover new phenomena. And so I'm really interested in whether or not there are universal laws that apply to biology, that apply to what we call life, that don't just apply to life on Earth, but would allow us to have predictive frameworks for thinking about life in other places. And so the sort of history of how I think about the problem is really to think about these sort of deep foundational questions and how do we actually get at new understanding of the way that reality itself works, not just thinking, you know, we're looking for life on another planet, we're looking for cells or something like that, but sort of what is the deep explanatory framework. And so just to give sort of an example of how hard I think the problem is and how far we have to go, I'm just going to talk a little bit about the history of relativity, because I think this is a really interesting sort of microcosm of the relationship between developing theoretical ideas that are deeply explanatory and allow us to uncover new phenomena in the universe, but they also have to keep in step with technology and actually make predictions that we can build technology to actually interface with the sort of predictions that we're making. So, so this is obviously a picture of Albert Einstein, and a little over 100 years ago, he was formulating these ideas about the general and special theory of relativity, and he was able to make a lot of predictions about the way the universe behaves, which were, some of them were validated on a short time scale. So for those familiar sort of with the work, Eddington did an expedition just a few years after Einstein basically described the way our universe works and the way gravity works in terms of the curvature of space-time. So he had this sort of like really deep conception of things that he came up with as far as trying to explain gravity. It's not like we weren't trying to explain this phenomena for a very long time. So the explanation that Einstein came up with of, thinking about the properties we associate to gravity and the interaction of massive bodies as being related to this curvature of space-time was very deeply abstract. And he actually got at that idea by starting from thinking about a very different phenomena by thinking about the properties of light. And if we look at sort of planetary motions and things that are supposed to be responsive to this idea that he came up with of space-time, the things that are actually space-time is, describing the motions of, we did not think about it in terms of that kind of explanatory framework for thousands and thousands of years, even though we might have had descriptive or even predictive descriptions of some of those phenomena. So for example, in antiquity, people used to talk about the Earth as the center of the universe, and we actually describe planetary motion as being perfectly circular, and to actually fit the data, in particular, the retrograde motion of Mars. So Mars sometimes looks like it's moving backward across our night sky. We actually had to fit perfect circles to these motions. And so this epicycle model was basically built up as all of these perfect circles around perfect circles around perfect circles, because in antiquity, they thought the perfect circle was sort of the heavenly object and was the necessary sort of mathematical structure for describing the heavens. And that worked for thousands of years because it was really predictive, but it wasn't sufficiently general or explanatory to allow us to describe other features of our universe, and in particular to maybe predict how other solar systems might have behaved if we had knowledge about them, because obviously it was the wrong model and it wasn't explanatory enough to go beyond just describing the motions of our planets in our night sky. That kind of description obviously took many hundreds of years to develop. So first Newton came up with the idea of unifying terrestrial and celestial motion, which was completely radical to the ancients, right? They thought the heavenly sphere was a completely separate kind of existence from existence here on Earth. But by the work of Galileo and Newton, we came to realize that they were described by the same fundamental laws and principles. And then Einstein, as I was talking about just over 100 years ago, came up with this idea of space-time, basically by talking about the properties of light and trying to deduce what they actually would say about the sort of constraints on the geometry of space and time as we can talk about it. And so now when we talk about gravity as this sort of universal property of our universe, we talk in terms of this very abstract phenomena that is the curvature of space-time. So it's not like at all obvious to me that I'm sitting here right now because of the space-time curvature of the potential well in the vicinity of Earth, or to you, right? It's not directly perceptible to our senses. Now, what we did with that theory is something really incredible. We made contact with a phenomena in 2017 that we predicted existed, but we had never actually directly measured in our universe before. And I say make contact to be necessarily sort of provocative about the idea that we have yet to make contact with alien life because we don't understand that phenomena. So what this sort of history of gravitational physics is trying to get at is, when Einstein came up with the ideas for that theory, it was very abstract and it was very far ahead of its time. And it took us many decades of building this interferometer technology to actually detect one of the predictions that he made, which was gravitational waves. So this is just one of the detectors for the LIGO experiment. For those of you that know about LIGO, the individual interferometer arms of LIGO are about 4 kilometers long. And they're necessarily so long because the effect of gravitational waves that you need to measure in order to validate that you're actually observing that phenomena, because it's a very weak phenomenon in our universe, is about, the width of 1/10,000th of a proton or something. Is like the sensitivity of this instrument. Right. So, but we have very high confidence of the first contact with gravitational wave phenomena that we made, which was a black hole merger that happened about 1.4 billion light years away and led to this very, sensitive instrument picking up these ripples in the fabric of space-time, because we had this really deep explanatory predictive theory and we knew what we were looking for. So this phenomena of gravitational waves has been all around us for all of human history. We had no concept of space-time, no idea that there were actually ripples in the fabric of space-time until we actually came up with this explanatory framework and were able to make these kind of predictions. So relatively 100 years ago was sort of in the theoretical idea stage, and now it's become one of the greatest windows we have into the nature of how our universe works. So this is a telescope, right? It's not how people would have thought about a telescope 100 years ago. And so when we think about astrobiology 100 years ago, we were in a really different place. Astrobiology 100 years ago maybe might have been concerned with talking about Martians on Mars, which people thought might actually be there. And we were also talking about the origins of life and how we might even begin to approach it as a scientific problem. So this quote is from an essay by Haldane, who is very well known in sort of early origin of life work, and was part of, along with O'Perrin, basically proposed a sort of primordial soup hypothesis that if you have a complex enough mixture of organics on a primitive Earth, it might have just been the spark that was necessary to start life. So there's this sort of idea of this hot dilute soup. And so this is kind of a descriptive explanation of what might be happening. And it did lead to a lot of kinds of experiments that we could do, including Miller's synthesis of amino acids in the 1950s. But here we are 100 years later, and we haven't solved the problem of the origin of life. And I want to point to it as a specific kind of instance of how we should be thinking about the problem, because a lot of the way that we've been thinking about it for the last 100 years is that life is chemistry. And if we make the molecules of life under some conditions, we'll have solved the original life problem. But it doesn't seem to be that the problem is exactly that easy, because life is not necessarily the chemical components of life. It seems to have these sort of more abstract properties that we might want to associate to it. And so when I'm thinking about the sort of problem of what, as astrobiologists, we should be thinking about, if we talk about life as this universal phenomena, in the same sense that we might talk about gravity as being a universal phenomena, there's rules and regularities in our universe associated with that phenomena. When we get to that sort of level of description, when we can talk about that phenomena happening many places in the universe and the properties of that phenomena, usually we have to go to some kind of level of abstraction to talk about it. We never talk about gravity being about the rocks or black holes or the large scale structure of the universe. Gravity is sort of this abstract property that describes a lot of the regularities and the features of the things that we observe. And right now, I think when we're talking about chemistry, we're talking about some features we associate to life, but what are those sort of underlying abstract principles that we expect to happen in any living system on any planet? And it's probably not necessarily just the chemistry of life on Earth. And so this is starting to get into this what is life question, which is sort of the core motivator for me personally in the kind of science I do. I'm really motivated to try to understand the nature of life in the universe and how we can talk about it from an idea that's much more fundamental and allows us to derive properties of life. But traditionally, this is not really how the field has approached this problem. We usually think about life almost in a sort of a phenomenological approach, where we would just want to describe features of the phenomena or define life. And so defining life itself has many kinds of problems associated to it. So this is just a word cloud of sort of typical definitions that people will put when they're trying to define life, and then using that maybe to inform some hypothesis about the origins of life or some way that they're thinking about biosignatures. But all of these are plagued by similar issues. And just to give sort of one example of a definition for life, there's a wonderful essay that Carl Sagan wrote called Definitions of Life. It's like 2 pages long. I highly recommend it. It's really nice the way he kind of turns things around and plays with ideas in it. But here's a definition that he cites as sort of a common definition that someone might want to propose. And it's any system capable of performing a number of such functions as eating, metabolizing, excreting, breathing, moving, growing, reproducing, and being responsive to external stimuli. Certainly, probably a lot of us are doing lots of these things right now, even as I'm talking or you're listening. And so this is great. It seems very descriptive of the phenomenon we might associate to life. But what Carl pointed out in this essay is that if aliens were visiting Earth and that was their definition for life, they might think that cars are the dominant life form, because cars satisfy sort of all of these descriptors. And in fact, nowadays, they might actually really think that, because cars have even been to outer space. So obviously, they're spacefaring now, this life form on Earth. And so one of the points that Carl made is that even when you have this sort of like obvious definitions, there are these cases that seem to really contradict what your starting premise was. So he concludes, based on that definition in this example of aliens thinking cars would be the dominant life form, that many such properties are either present in machines no one is willing to call alive, or absent from organisms that everyone is willing to call alive. And I would actually not draw the distinction at machines, because I think all of our technology is as representative of whatever life is as we are. But I think his point that if you start from a reasonable set of starting assumptions and you want to make a definition for life, you always end up excluding cases you intended to include and including cases you tended to exclude. And the point of highlighting this is that life is not necessarily intuitively obvious to us. When we make definitions for life, we have an intuition and a clear idea of what we want to include, but we always end up with these contradictory cases, which is suggesting to me that our intuition and our sensory perception of what we are talking about as far as the phenomena we're aiming to describe is not exactly accurate to reality. It's not possible to actually define life, because there's no boundary that is a direct, this is life, this is not life. That's not the way that problem needs to be structured. And so, if you want to kind of push that kind of perspective to the far end of the spectrum, the sort of citation for this top quote has fallen off, but this was something that was said by Andy Ellington at the 2012 American Chemical Society meeting. I think he still holds this view, but very radically, puts up on his slides, life does not exist. Let's move on with solving the origins of life. Jack Szostak is another prominent origin of life chemist, and has this nice essay about how definitions for life will not help solve its origins, where he says, as one focuses experimentally on any of the defining properties of life, the sharp boundary seems to blur. So basically both of them are pointing out the fact that when you get down to the molecules of life, it looks like life doesn't even exist. So why are we worrying about defining this property we call life? Let's just move on with solving the origin of life. I always found this kind of hard to take, because I think if you're talking about the origin of a phenomena, you have to have some idea of what that phenomena is. And so my personal approach to the problem is really to go back to that sort of abstract principles, which is why I was giving the gravity example at the beginning, and really think about the nature of the problem that we're trying to answer, and how deep of a description or how much of an explanatory framework we really need to be thinking about. And this is sort of the approach of physics to just nature in general. So physics is, traditionally thought of as studying things like gravity or particle physics, but it's really just sort of a way of thinking abstractly about the reality in which we live, and trying to come up with sort of first principles explanatory frameworks that are very encompassing of a large set of systems and explanatory of them. And so one of the people that was really prominent in early thinking about the nature of life from this perspective is Erwin Schrödinger, who's a famous quantum physicist, who wrote this wonderful book called What is Life?, where he asks basically the question, what is life? But toward the end of the book, he kind of, goes through all these sort of arguments about how life is consistent with known laws of physics and things, but basically says we can't explain it based on those laws, and we really should be looking for other laws of physics that we currently just don't understand. And so there's no reason that we can't actually have an explanatory framework for life just because we don't have one yet. So this is a quote from Albert Einstein, just on the premise of theoretical physics. We should be able to explain any natural phenomena whatsoever, including life, by means of pure deduction. Now, you could debate whether that's true or not, because a lot of people think that life is not a natural category, or life doesn't exist, or it's an epiphenomenon, and there should be no fundamental explanation. But I think that's not consistent with what we're trying to do in astrobiology, where we assume there is a phenomenon in the universe we call life, and we're out there looking for it. We have to assume that it has some universal foundational properties to it. And so that's sort of my starting point. But the way I approach the problem is to think about it much more like the laws of gravitation. There's some universal physics that underlies this phenomena we call life, and we need to identify what that physics is. And then the features we associate to life will be able to pop out of that sort of more fundamental understanding. And it's sort of like we acknowledge gravity exists universally, but when we study it, we study these sort of massive bodies, we study planetary formation, or we study black holes, or large scale structure of the universe. Those are examples of gravity. We don't go to nuclear physics to study gravity, because gravity is pretty irrelevant inside an atomic nucleus. What I think is sort of the most promising approach to thinking about this foundational theory for life is to think that there's this thing called information, which is very prominent in living systems. And so it's sort of the dominant physics in life, but it's not dominant physics in other areas of physics we've studied so far. And that's one of the reasons that physics has remained hidden in sort of the way we've talked about physical systems in the past, but in life, it's very prominent. And so when I talk about information, I'm not talking about information in a Shannon sense. I'm talking about information more in a causal sense, in the sense that information like knowledge of the laws of gravity, which is a regularity that exists in the world that our biosphere has learned over many billions of years, but in particular has really learned to compress in a mathematical understanding of the world in the last few hundred years, is directly responsible for our planet's ability to launch satellites into space. We wouldn't be having a space program and launching little metal boxes into space unless we had knowledge of the laws of gravity. And likewise, our technology and our knowledge of nuclear physics is directly responsible for certain elements being produced on the surface of our planet right now. And so these are very extreme anthropocentric examples, but they're just meant to highlight this idea that if you want to understand why certain things exist, like satellites orbiting our planet, or high atomic number elements being present on our planet, or a protein, or a DNA structure, you have to have information somewhere in that sort of explanatory framework, or some fundamental idea of what information is as a physical system. And so this kind of gets to sort of the core premise of that motivates a lot of my work, which is just thinking that the origin of life transition, when we're thinking about it, is not just let's make a cell and get, Darwinian evolution started. It's actually a much more fundamental transition that happens in physical systems in terms of information not mattering much to how those systems behave or act or what they do, to information being the dominant physics. And so I make a kind of, I stand on my soapbox of like, life actually represents new physics. And that may or may not be the case, but I think this sort of way of thinking about it and framing it is incredibly promising as far as the number of new ideas and new tools it gives us for thinking about astrobiology and the way we do our science. And so most of the rest of my talk is going to be talking about some of the implications of that in terms of like the kind of technical work we can do, not the philosophy, but just adopting this idea that life might be a manifestation of a much broader and deeper explanation than we've traditionally given it credit for. And so sort of the picture that gets built from this idea is really that when we're thinking about the evolution of life on Earth, life is really sort of this process of information structuring matter across space and time. It happens, in the origin of life on a planet. And then basically you have this sort of cascading evolutionary process building all of these structures that we call life, and then technology. And then eventually planets might even reproduce themselves by sending technology off world. And it's much more of a continuum. So it's not this thing's life, this thing's not life, but there's degrees of being alive in life. And this life not equal alive means that some things are very active participants in this process. So I, for example, would consider myself alive. My cup is also a product of 4 billion years of evolution and would not exist in the universe unless our biosphere was here. But my cup is not alive, it's life. It's evidence of life, but it's not itself an active participant in that process of information processing and this kind of idea of building structure and complexity. So just to re-articulate that, the origin of life transition is really finding this transition from non-living to living physics. And if we understood this living physics, it would deeply inform how we should be thinking about biosignatures, because it allows us to now not just look for things that happened on Earth to happen elsewhere in the universe, but to actually make predictions about what should happen on a planet based on its particular geochemistry as it enters this sort of evolutionary cascade of information, basically producing more structure out of that planetary context. So I'm going to talk sort of in the last part of my talk about the theory that's sort of underlying how we're thinking about these ideas, which is really sort of where I like to do science is, sort of giving the philosophical background, but actually doing the work like directly at the boundary of theory, experiment, and observations, because I think that's where we have the most scaffold for those kinds of ways of doing science to scaffold off of each other and actually build a paradigm where theory is directly testable and experiments are actually corroborating and helping build the theory. And so I'm doing this with a lot of collaborators, but a key collaborator is Lee Cronin's lab at University of Glasgow. And my group's a theory group, his group's an experimental group. And together we're trying to develop this idea called assembly theory, which is basically trying to talk about the physics of life from the perspective of thinking about the complexity of objects as being generated by information. And I'll talk about that a little bit later, but I just wanted to say that there is a direct connection between this sort of abstract idea and things that we measure in assembly theory as far as the complexity of molecules and how we think about the causal structure of living systems as being generating this sort of possibility space, and that certain things like nuclear physics wouldn't happen without this sort of chain of events that had to precede it as far as evolution happening over a planet over 4 billion years. Okay, so that was looking 100 years ahead. How do we get theory to actually, make astrobiology equivalent of an interferometer in 100 years? But we have to do the theory work first, in my mind, or at least that's how I'm approaching it because I'm a theorist. And so there's a lot of things that we can actually think about concretely about how to build into this sort of set of ideas about this sort of deeper philosophy about the nature of life in the universe and how we can think about alien life and also our own origins. And so my group is approaching that from a lot of different directions, but we have like sort of 4 major pillars. And I'm going to talk about sort of 3 of them today and not the other one. But the first one in which I'm going to talk about probably the most is statistical approaches to characterizing life's chemistry, which is this idea of planetary systems biochemistry introduced by Rory at the introduction, which forms sort of foundation of our ICAR project. And then how we can think about universal signatures of life, both with thinking about these statistical properties of living systems, but also assembly theory. And then this kind of physics of information and causation, which is sort of the foundation of the theory that we're building. And I'm not going to talk about the nature of intelligence because that's all, minds are a whole nother beast entirely, but they're related. And so a concept of really important for astrobiology is this concept of universality. And so we talk about universal properties of life and how we can look for them quite a bit with respect to the chemistry of life. And so the chemistry of life, at least on Earth, is universal in some respect. So, we have this idea I mentioned at the beginning, which goes all the way back to sort of, even 100 years ago when people were thinking of astrobiology about life as chemistry. And one of the key features of life as chemistry when we look at it across the Earth is that all organisms and all life forms that we've ever identified share basic component parts. So things like amino acids and RNA and DNA and all of these sort of core biochemical components are universal across the tree of life. And this is one of the reasons that we can even talk about the idea of a last universal common ancestor or common ancestry for all life on Earth, is because of this sort of shared biochemical component parts. So there's this sort of idea that we expect life to have universal features. So it might be that those features are in the generality, if not the details. So are these particular biochemical components actually the ones we should be looking for elsewhere, the particular metabolisms we find on Earth, the particular features, the particular component parts, or is there some more general concept of universality that we could derive from the chemistry on Earth that we see? And so what kind of more general idea of universality would you be talking about? Well, in physics, we have a very different notion of universality than we do in biochemistry. The idea of universality, as we talk about it in physics, is this idea that systems behave very similar in some kind of aggregate or macro scale sense. So a good example is sort of the physics of phase transitions, where you might have two very different physical systems. So their component kind of, base level physics is really different, like the liquids and gases or ferromagnets. But when you actually look at their phase transition behavior and you look at how certain properties of the system scale with, for example, as temperature, you see similar scaling coefficients. And then we talk about the phase transitions or the behavior of those systems being the same in some universality class. So they're described as basically the same universality principles. They have the same scaling behavior, et cetera. So that's different. It's a macroscale feature, not a microscale feature. It doesn't depend on the component parts like it does in biochemistry. And so one of the questions my group has been asking is whether or not we can talk about universality in the physics sense, but apply it to biochemistry. And so this is really sort of the sort of starting point for thinking about this idea of planetary systems biochemistry, that we might think of Earth not in terms of the details of the specific kinds of biological systems that exist on Earth or the kinds of chemistry that they use, but we might abstract it to try to actually look at statistical regularities in the chemistry that life uses and how they're driven by planetary environment, and then build basically statistical frameworks for predicting what other kinds of chemistries we might observe on other planets with different geochemistry. And so the way that we do that is actually to talk about scaling behavior right now, but we have other methods that we're thinking about doing. And this kind of scaling we're looking at is to take a biochemical system, which might be an individual organism or an ecosystem as you get data from like a metagenome or something. And then you look at the biochemistry, all of the reactions that organism can catalyze with its enzymes. And then you say there's, the size of the system is the number of compounds implicated in that biochemistry. And then there's some property that we're looking at scaling behavior with. And the sort of first work we published on this was out in Science Advances a couple of years ago, looking at network properties, which was actually looking at the statistics of the patterns of the interactions of the molecules in actual reaction networks. So this is an image of the actual whole biosphere, all the reactions we know that biochemistry does, but we looked at individual organisms and ecosystems and demonstrated that networks had scale invariant properties. I'm not going to talk about that work today. I'm going to talk about some newer work, but the foundation of this idea of doing statistical sampling is the same. And so what we do is we basically try to aggregate as much data available in databases across Earth as possible. So some of the databases we use are things like the Joint Genome Institute, which catalogs thousands of genomes and metagenomes. The data list that I have here is actually undersampled compared to what we have now. So I think there's something like 60,000 data points in the database that my group has been curating at this point. But the idea is when you get this genomic data, a lot of it's annotated with function. There's a lot of issues in those pipelines for people that work in microbiology. We know it's not like a complete map of function, all those kind of things. And I love talking about those kind of details and how we can build better tools for inferring properties of microbial ecosystems in addition to the kind of work we do. But the sort of main premise is once you get a large enough statistical sample of what biochemistry is doing across planet Earth, we can start to actually build better tools for understanding these regular properties, right? Statistical regularities, and building kind of law-like principles for why life uses the chemistry it does. So one way we've been doing that is actually to look at enzyme functions. So enzyme function happens to be cataloged in a particular way that's actually very useful for this kind of work, which is it's cataloged hierarchically. So when you get an enzyme and you assign a function to it, you have a 4-digit enzyme commission number that people will assign. So for example, 1.1.1 is the function alcohol dehydrogenase. But if you start looking at sort of less specific digits, enzyme functions get grouped together by common functions. So by the time you look at the highest EC level digit, the first digit, you're in a pretty broad class of enzyme functions. And it turns out that there's actually sort of 6 major classifications that people have used. There's actually a 7th now in these databases, which is translocases, but we didn't use them because they're too new and not under annotated. But if you look at these 6 broad functions, they're kind of like a coarse graining in the sense that we use in physics. They group a whole bunch of things that have similar properties together, but they group it by function. And so what we wanted to know is if we looked at biochemical reaction space, just with respect to these 6 very broad functional categories, could we actually extract any kind of universalities or statistical properties that might allow us to understand how biological systems function and what their statistical regularities are, independent of the specific details of reactions and molecules. And so to do that, we basically went into those large ensemble databases and we looked at every enzyme function in every genome and metagenome, and we counted how many redox functions there were, how many transferase, how many hydrolase, and then basically plotted for each genome or metagenome what the ratios were of those to the total number of enzymes. So how many of each function do you have relative to the total number of functions in that biochemical system? And when you do that, we get very regular scaling behavior for all of the enzyme functions across all of the data sets. And so this is suggesting there are statistical regularities in biochemistry associated to these very macroscopic features of the functional classes of the reactions that biochemistry uses. That could be potentially predictive of other biochemistries, but also missing biochemical diversity in our biosphere. And these are the particular scaling fits that we had in the paper. And so certain enzyme classes like the redox reactions and hydrolase reactions are superlinear in their scaling fit across all data sets. Some of them like ligase and transferase are sublinear. And so when these classes are actually have a fit that's shared across all of the, like a fitting behavior, that they either scale super linear, so you get enrichment of that particular class with respect to the size of the system as the system grows. So redox, you end up in large biochemical systems that have a lot of redox and very few ligase. If that behavior is universal, we said that that's a good candidate of a universality class. It turns out that lyase is this kind of funky one that doesn't seem to have universal behavior, but lyase function is identical to hydrolase, except you've separated out hydrolase because you're actually using H2O and for the function as a specific kind of molecule case study. If you group these, then you get another universality class with a tighter fit that is just degradation reactions when you break molecules apart. And so in the actual manuscript that we have, which is in final stages of revision review for publication, we actually ended up grouping these, and then we talk about 5 universality classes. Isomerase are another sort of outlier, but this is actually statistically consistent with possibly having a sublinear universal fit. So there seem to be 5 classes of function that have universal behavior across all biochemical systems, across all scales, with very well-defined scaling fits that allow us to talk about this sort of idea of a macroscale universality class in biochemistry. Now, one question you might have is, well, we know life on Earth shares universal component parts. I said that a few times. That's a really important detail. So one of the things we had to do was actually check and make sure that the scaling fits that we have and this kind of behavior that we see emerging in these large scale trends across biochemistry is not a direct consequence of the fact that there are some reactions that just happen in every organism. And so to get at that, we built these sort of universality score statistics. And the way to read these plots is if you're looking at the x-axis and you're at sort of 1 on the x-axis, that means that that enzyme function is found in 100% of that data set. So I'm looking at the orange line on bacteria here, which orange is for ligase function. So this enzyme function here is found in almost 100% of our data set for bacteria. And then you basically just rank order all of the enzyme functions by what percentage of your data they're found in, and that basically generates this curve. So things that are flatter and higher, so have a higher rank order on the y-axis, are found in more of the sample. When you're talking about things having high universality, they would have a very high area under the curve score. So this area under the curve, which is in this table down here, is basically just how flat is this curve to 1 across the top. If any of these numbers were 1, it would mean every enzyme function in that class would be found in every member of that data set. So the highest sort of scores we see are for ligase functionality, which is not entirely surprising because we know ligases are implicated in sort of related to like transcription and translation machinery and sort of the functionality of like core biochemical functionality. A lot of ligase functions are found in common across a lot of organisms and also across a lot of metagenomes. So there's a high universality score for that function. By contrast, redox enzymes are very low universality because a lot of redox functions are unique to a particular organism or a particular ecosystem. So we find a much lower universality score for redox function being distributed across the biosphere. So then when we look at the data like this, we can talk about the fact that metagenomes have more universal components than individuals, and also that some enzyme functions are more universally distributed across life than others are. And this gives us sort of a statistical picture with respect to these functions of how distributed they are. Now, we can also look at how are those AUC scores, those universality scores, correlated with scaling coefficients. And the basic point of this plot is just to show that they're not. So basically, we've identified these sort of macroscale patterns in terms of scaling patterns and these microscale patterns in terms of universal components. So this is biochemical universality, and this is physical universality. And there's no direct correlation between the two when you actually plot one versus the other. And in fact, it's super interesting because oxioreductases have the tightest set of scaling fits, and they're the least universally distributed. And ligases are more universally distributed, but they have a wider variation in their scaling fit. So it's not even that they're not correlated, it's that they show sort of the opposite trends of what you might expect. The things that are more physically universal are less biochemically universal. So that's sort of one of that sort of corroborates our idea that maybe we can start thinking about these statistical patterns of biochemistry and actually separate this idea that statistical patterns exist from the underlying particular features of the biochemistry we know on Earth. And what we've been trying to do is basically get at the idea that we can derive that from some sort of fundamental theory or functional principles. So we're working on the theory now of basically how to build simple models that recover our scaling features and allow us to make predictive frameworks for alternative biochemistries for life as far as what we would expect for their functional diversity and what kind of chemistry they could use. But before we can even get to that, we wanted to try to look at the structure of biochemistry as we know it. Because I'm going to be sort of short on time, I'm going to just talk about the plot on the left, which is looking at sort of starting from, if you look at that biosphere level network I was talking about before, and you start from primordially available compounds, there's sort of a standard set of compounds that people use in this sort of network approach to thinking about the biosphere. And you start growing biochemistry by iteratively adding reactions that you now have available resources to conduct that reaction, assuming all enzymes are there. You can basically track how enzymes were added sort of at this very coarse level. And obviously that doesn't recapitulate evolutionary history, but it tells us where current enzyme functions are relative to sort of this core where biochemistry might have started. And what we see is sort of the sort of sublinear classes peter out really quickly. They achieve all of their diversity very early in biochemical network expansion. But what we see is much later sort of transferases and oxioreductases, which are like these super exponential, super linear classes, are still growing and adding more diversity to the biosphere. So if you think about the evolution of biochemical diversity in the biosphere as a whole, there's certain functions that dominated very early and are no longer contributing much novelty to our biosphere. And then there's other functions like redox that continue to be very innovative. And it's very easy to see that sort of in the structure of these systems. And so now we're kind of interested in tracing that back in time. So we have this sort of statistical imprint in all of this data we have on Earth today about these features of biochemistry, but can we actually trace them back to the earliest signs of life? And So one of the things we're interested in doing is taking these scaling laws and trying to statistically constrain the properties of the last universal common ancestor. So a lot of people propose this idea that the last universal common ancestor might be some phase transition in this collective property of early life. But we have to reconstruct that in the details of the specific chemistry based on current phylogenetic reconstructions. But I think what we can do with the scaling laws is actually talk about the properties of that phase transition in chemical space, as far as, we see these very regular statistical patterns in life as it exists today. If those are universal to all life, they must have emerged when sort of the first biochemical system solidified on the early Earth. So we've been starting to work with this a little bit with thinking about sort of where current reconstructed models of LUCA fall with respect to our scaling laws and seeing some variation there that might allow us to sort of build new ensemble statistical models for the last universal common ancestor, which is a current project in our group. So I really do want to talk about assembly theory. So I'm moving a little fast and I'm sorry about that, but I guess I put too much stuff, but there's always so much fun stuff to talk about. I'm going to talk briefly about symmetries just for a second, which is to say, I've been talking about universal features in reactions, which are transformations among molecules, but we can also look at the patterns in the individual molecules themselves, which is sort of a big area of research in a field called cheminformatics, which has to do with like pharmaceutical drug design and things like that. And we use a lot of the same statistical tools that they use in the pharmaceutical industry to do this. But one of the features we might look at is molecular chirality, which for those of you that are familiar, is an important sort of feature of symmetry breaking in the origins of life, that we only see left-handed amino acids in proteins, but we can prebiotically make left and right-handed amino acids, for example. So for a long time, people have focused on this idea of the handedness and this breaking of symmetry, that one hand exists now and the other is, kind of gone from biochemistry. What we're doing is a little bit different, which is to think about chirality itself as like a property of every molecule. It's either a chiral molecule or it's not. And if it's a chiral molecule, it has a certain number of chiral centers. And then we can think about the statistics of that feature of biochemistry and not even look at sort of macromolecules and their homochiral features, but as sort of like a more primitive way of looking at just in sort of small molecule chemistry, the chiral signatures of life. And so to do that, we have to take our entire database and basically label the molecules as being chiral or not, which this is kind of showing what the biosphere looks like with respect to chiral versus non-chiral molecules. And if you look at prebiotic chemical reaction synthesis, there's some scaling that you might expect where the number of chiral compounds increases as you're increasing molecular weight and as increasing number of diversity of compounds. And so people kind of see this feature in prebiotic chemistry. But what we see in real biochemistry is we do have very rigorous statistical scaling laws with respect to the ratio of chiral molecules to non-chiral molecules in the biosphere. But chiral molecules are actually sublinearly scaling. So as you increase the size of the biochemical system, you're actually getting a enrichment of small molecules that are achiral. So this is another scaling law that we're looking at and studying sort of the features and demonstrating that, these are not features that you would a priori predict just from the behavior of molecules and their relations and reaction networks that aren't biological. It seems to be very distinctive to the particular features of biochemical organization. Okay, so in this sort of last part, I'll spend like 5 minutes on this last part and then open for questions because I want to make sure we have enough time. Is thinking about sort of the principles that will explain life. So to go back to the introductory stuff. So some of these features I'm talking about in terms of networks and statistical patterns are ultimately aimed to connect back to these fundamental ideas about the nature of life. Because I think when you build the theory, you have to build the biosignatures and then you have to gradually start converging the theory and the biosignatures. So I think a lot of the patterns that we're talking about will ultimately be predicted by this sort of assembly theory framework that we're building. And so I'll talk a little bit about assembly theory. And it's really coming out of this other Einstein quote that I love, which is one can best feel in dealing with living systems how primitive physics still is. So our research paradigm that I'm working on with a number of other people is really to come up with a new understanding of physics, which is very different than the physical laws and the kind of physical theories we built in the past, that actually is explanatory of life. It's a very sort of deep and encompassing research program. And I'm optimistic about some of the features that we're seeing because what we're trying to do is start from observables to build theory, whereas most people are really thinking about building a theory for the sake of building a theory and not really tying it to observables. And the observables that we're talking about and the kind of theory we're trying to think about is to think about sort of the space of all possible molecules and all possible objects that could exist in a biosphere. So for example, when you get to technological space, that space is exponentially huge. But even in small molecule chemistry, the combinatorial space is so large that we can't make every possible small molecule. I think there's estimated like 10 to the 60 molecules with a molecular weight less than 600 Daltons. So, I mean, these are exponentially huge. So I have a picture here of the Hubble Deep Field versus an image of chemical space, just to demonstrate that people think the universe is big, but the universe of possibilities of what could be generated on a single planet with life on it, as far as the complexity and the combinatorial space of things that could be generated, like chemistry or technology, is so large. If you can compare a combinatorial space to a physical space, it would vastly outweigh sort of, you know, the degrees of freedom in the Hubble Deep Field, for example. So it's huge. And the other sort of feature I just want to say here is that, you know, life is actually tied to planets. There's a reason that life emerges on planets, which I think has to do with the fact that you get a high enough density of things happening in this sort of combinatorial space that life actually can emerge from the features of chemistry on planets, but it can't emerge in, say, an interstellar gas cloud. So it is something that happens on planets, and we have to take that into consideration. So this planetary contingency is really important. I'm not going to talk about it in too much detail on this slide because I have another one that I can talk about. So the premise of assembly theory is to talk about mapping this space of all possible molecules in a very systematic way, where you talk about a molecule, you break it in half, you break it in half again, you break it in half again, and you basically construct all of the ways of assembling that molecule from elementary building blocks. In this case, the elementary building blocks are bonds, and that defines an assembly space for that molecule. So in the theory we're building, everything that exists is an assembly space. I'm an assembly space, a molecule is an assembly space. And we can talk about how much evolutionary history is created in the universe as a minimal bound to produce that object as the shortest length in that space. So when you start from elementary building blocks and you're building up an object like ATP, there's an exponentially large number of possibilities as you're adding more and more structure to the molecules. So by the time you get to ATP, there's a huge space of possible ATP-like molecules. But ATP is a very specific one. And ATP happens to have a molecular assembly index of 21. So it has a pretty long number of steps to produce ATP. And so what we've tried to do in assembly theory, as far as building this into a biosignature, is to talk about that assembly index, that minimal path in the space, as the biosignature. And if that path is above a certain threshold, then we assume that there had to be a system that had information about how to produce that object. There had to be an information processing system, or a system that had evolutionary selection to produce that specific object, whichever way you want to talk about it. We don't have the physics clear yet, so we talk about it a lot of different ways. And so then this becomes sort of the foundations of a life detection system, because you can take non-living systems, dead systems, biological systems, and then also, so this experimental work was done in Lee Cronin's lab at Glasgow, as I mentioned. You can also take blinded samples that were sent from NASA to his lab to actually test this framework and determine if it works. And so the first point I want to make is that assembly index, even though we mathematically derive that from the structure of the molecule, the first thing that had to be done was verify that that's actually an observational property of a molecule. And so for me, this is the real key thing, and one of the reasons that I started really wanting to work on this framework in a really rigorous way for thinking about all of these sort of abstract properties of life, because assembly index is actually observable for molecules. And the way that we do the observation is we make a prediction of the assembly index based on the actual structure, the graph of the molecule. And then you can go into the lab, and with a mass spec or an NMR, an infrared instrument, you can actually directly correlate that with features that you observe in the molecule. So in a mass spec, you, for example, can correlate the assembly index with the fragmentation pattern you see in the mass spec, and you get a very strong correlation. And then you can say that mass spec is actually allowing you to infer the assembly index of a molecule, even if you didn't know the molecular graph and things. So this is sort of the foundation of it. And then when you go and you take a whole bunch of different samples and you use mass spec on them, you can basically predict what the assembly index should be for those samples based on this strong statistical correlation. And so this is sort of the, so the estimated MA and the sort of mass range of the molecules, and then what we see for the different samples that were tested. And these are just like some specific case studies, but basically what you see is above the sort of molecular assembly index of 15, which is a little clearer in this sort of smooth data I'll show on the next slide. Above 15, we only get biological samples producing high molecular assembly objects, which is kind of consistent with this theoretical idea we had that if you want to see a high complexity object, you have to have a system that knows how to produce it, because the combinatorial explosion is just too huge. And so one of the key features of the mass spec instrument in particular is you will not observe a molecule in mass spec unless you have more than 10,000 copies of it. And so the sort of boundary at 15 was predicted even theoretically, because if you get to an MA, a molecular assembly index of 15, and you're doing this sort of random exploration over this combinatorially exploding graph, you would expect to have less than 1 mole copies of that molecule. So you would never detect it in your mass spec. So when we see it in the mass spec, it indicates that there's some reliable mechanism that can produce at least 10,000 copies of that molecule, which is indicative in this theoretical framework of life, because life is sort of the hypothesis that life is this information processing system, the system that has specificity about specific objects that could exist. So that's sort of the foundation of assembly theory as being derived from properties of molecules that are observable in the lab. And what we're doing now is basically trying to build that into this new physics-based approach to thinking about life, where every sort of thing that's generated by the biosphere, or even in physics, an electron has an assembly space, a molecule has an assembly space, I have an assembly space, I am an assembly space. But based on sort of the shortest path in that space, there's kind of an ordering to how objects can form in time, and also what objects have to precede other objects, because they're part of this causal structure that's generating things. And so you can see kind of how you have this sort of underlying explanation for what evolution is in terms of the causal structure and the causal relationship between these objects and assembly spaces. And so right now we're working on some theory on statistical mechanics in assembly spaces, which is basically kind of a refinement of statistical mechanics, but actually accounting for history and structure of objects that we can observe that are specific features of life. And so that's sort of where that work is going right now. And aliens, how do we recognize life? I just want to say that, like, all of this statistical approaches and trying to build assembly theory is really for this kind of vision. And Lee and I have been talking about this a lot over the last few years, about trying to build a large enough original life simulator to simulate planets, and then to be able to actually statistically explore chemical space on planets to look for de novo original life events in an experimental system. So like, imagine CERN looking for new physics, but doing that for origins of life. And so the technology exists to do that. And a lot of it actually has been developed in Lee's lab with this digitization of chemistry and this building this abstraction of this computer that basically allows you to make reaction space programmable and have robots doing parallel experiments, exploring all of these different conditions and generating data about sort of the statistical distributions and the products you might get in different environments. So this kind of framework is done, but demonstrating how it can couple to geochemistry and how it can build toward this sort of larger original life experiment is not something that has been done. And so this is not an original life experiment, but this is what I want. Someday I want us to have one of these for astrobiology, but this is Super Kamiokande, which is in Japan, and is this huge experiment looking for proton decay, which is a very rare event if it happens at all in our universe. So we have some theories that predict it, some theories that don't. And so basically we're trying to bound the lifetime of the proton by looking at this experiment and not observing events. Every time we don't observe events, we're basically saying the proton lifetime is, at least this half-life. So imagine we have like a huge experiment basically of these robots that can do these chemical synthesis experiments, explore chemical space to look for life. We know life at least happened once, so it has to be not a zero probability. So we could build the experiment. What we're doing as part of our IACAR is building the technology to maybe help build the foundation for this kind of experiment, which is the geocomputer. So we're taking the computer technology, which basically allows you to do these parallel experiments, but do it under geochemical conditions. So this is a collaboration between Lee Cronin's lab and then us here at ASU with Everett Schock and Hilary Hartnett, basically building this to start statistically exploring what kinds of patterns in chemistry we get under different geochemical conditions, and starting to build up to these sort of statistical frameworks that I was talking for life, but doing it in geochemistry and having the data to back it up. And then, molecular complexity obviously is, assembly is almost ready to fly on NASA missions. So we're hoping that maybe we can do that. The mass spec we've used for it so far is very high resolution, but it's a technological hurdle to get it to lower resolution mass spec, not a conceptual or foundational one. So we think it'll work for that. And then thinking about exoplanets and things. I know there's a lot of people that work on that here. I didn't talk about it too much, but we're doing sort of, thinking about these statistical patterns in planetary atmospheres as a biosignature. And my student Tessa Fisher has been working on this for a while, has some stuff on hot Jupiters, but has recently been working on terrestrial atmospheres. And this is so for planets with methanogenesis and not methanogenesis. And she's been using some sort of standard model in that, trying to look at sort of how different network features might distinguish these planets, even when the abundance distributions for a particular molecular biosignature might not. And so we're trying to work on building these statistical frameworks there. This is just a plug that I like talking about this stuff to people, but didn't have enough time to put everything in the talk. And then I'm just going to end there and thank my group. Hanju Kim has been absolutely critical to all the Planetary Systems Biochemistry. She's our deputy PI for the ICAR project, and she's absolutely brilliant and building new fields of science every day. And then our students that have been really involved on the projects I discussed are Veronica and John Malloy and Bradley and Dylan and PLR and Kamarian and Cole is also awesome. So thanks. And I thank everybody, and I hope I left enough time for questions. I know that was a lot to cover, so thank you.
Great, thank you very much, Sarah. So hopefully everybody can plug in whatever way they can in this virtual world. So we do have a few minutes for questions, and in fact, some people have typed them in to the chat. So I think the first one here is from Wallace Marshall. So Wallace, if you'd like to turn on your camera and ask the question directly, you can, or I can read it. So turn on your camera in a second. I'll let you do it. But all right, I'll ask the question. So he asks, how do you quantify what type of physics is quote unquote dominant? Is it a question of what processes use the most energy? So information is dominant physics equals life is where most of the energy is consumed processing information.
So I like that perspective. I've never thought about it that way. I usually think dominant physics is most explanatory at that particular phenomena that you're looking at. So for example, like when we're thinking about Brownian motion, we don't use gravitational physics, we use quantum physics. But so that's what I mean when I say dominant mode of explanation. It's just the one that's the best explanatory framework for that particular class of systems or that particular slice of reality that we're looking at. But I think this way of thinking about it in terms of energy is actually kind of nice. And it would be interesting to see if that actually plays out. And it also helps because people think life is a non-equilibrium process and just about energy consumption. And I vehemently disagree with that perspective for a number of reasons. But that's kind of a nice way of highlighting it because all physics uses energy. It doesn't mean it's uniquely explanatory. So yeah, that's the short answer.
Great, thank you. Next up is from Brooke Nunn, and I see she has turned on her camera, so I think she's going to ask you directly.
Hi, Brooke.
Hey, Sarah, that was an excellent talk. Yeah, so my question was related to like part 2 of your talk, part 2 of 6. So for the different enzyme classes that you had that you associated to these different domains of life, did you ever examine the ratios of those enzymes to one another for those different domains to see if there were like, significant ratios that were associated with a type of domain?
Yeah, so I think there probably are. What we're doing right now, and this is something Veronica is working on, and I think she was hanging out at UW for a little while, so some of you know her, but she's amazing and brilliant. But she's basically trying. So I showed the domain level data, but we also have data at the lower taxonomic categories. So you can like look at phyla, for example, or something. So basically what we're trying to do is map the scaling laws to the phylogenetic tree to try to see if there's consistency with variation in the scaling behavior with evolutionary divergences. And then we could basically predict particular sort of macroscale features of how biochemistry has evolved along particular trajectories. And I think what you'll see is that, there are legitimate size effects. So archaea tend to be smaller than bacteria, tend to be smaller than eukaryotes, tend to be smaller than metagenomes. So just at that scale, because we see these scaling laws, you expect different ratios of the different functions in those systems just because of the size effect that archaea have fewer enzymes than a community does. But whether there's sort of more nuance to that based on phylogenetic relationships and these sort of evolutionary constraints we're trying to figure out.
The other thing that would be neat that I think would be easy to add in would be to see if you can classify the organisms based on their origin, like soils, water,
yes,
biofilms, etc.
Yeah, so I did not even, there's too much cool stuff happening in our group. I don't get to talk about it all, but Pilar Vergelli has been working with metagenomic data for a while. So she has like about 11,000 samples and they're classified by environment of origin. And so we're basically trying to correlate, but what she's working on is more elemental distribution patterns. So I didn't show it, but we also have scaling laws for element use in molecules in life. And she has that data from metagenomes and try to correlate it with environmental context. And Kamarian Millsaps is trying to work from the other side of building the proper metadata for doing that inference. So we're trying to build the bridge between being able to predict some of these statistical patterns based on environmental data and how we can do that. So I think they're there, but it's really hard to extract those features from the data we have. So we have to build new data.
Excellent, thanks.
Yeah, it's a great question. Thank you.
All right, I was muted. Before I move on to the next question, I just want to make sure everybody knows it has reached 4 o'clock here in Seattle. So if you have someplace to be at 4, you can hop off, but we'll keep going here for anybody who's still here. The next question in the chat is from Alan Caswell. So Alan, if you'd like to ask directly, now is the time to turn on your camera. All right, not seeing him, I will ask myself. So he says, any possibility that the predominance of oxidoreductase processes in the result of our current oxygenated environment, i.e. great oxidation event, et cetera, or that the less dominant processes, ligases, et cetera, might be more dominant in a less oxygenated system?
Yeah, I think that's a super interesting question. I have reason to suspect that maybe that's not necessarily like the sort of naive hypothesis that it might not necessarily be correct, but it definitely is within the realm of possibility. But there was some work done by Daniel Segre and Jason Raymond, this like beautiful science paper in 2006 on the effect of oxygen on the evolution of complex life, where they took like all of the known reactions at the time, and they looked at this sort of network expansion algorithm with and without oxygen, and they showed how much a biochemical space can be reached with oxygen. So we know the network got a lot larger, and certain properties of it are definitely an artifact of oxygen being present, even if they're not oxygen related reactions. But I think what we see with sort of the expansion I was showing data for is the oxidoreductases start increasing their slope like pretty early. So this exponential ramp up they start to have is probably before we would start to see the effect of oxygen on that network expansion. But we haven't directly checked that. So I think we should. I think it's a good idea. But I think it's more of an artifact of like, when you build larger, it's more of sort of an energy argument for evolution. So I think ligase functionality is necessary early on because you have to basically be able to make biochemical parts, and you have to be able to compose them and to build molecules, right? So that needs to happen early, but most of the later evolution of life is about acquiring sufficient energy to build biochemical systems or to build larger biochemical systems. So it's not just that we see the oxidoreductases emerging later, but that we see more oxidoreductases in larger biochemical systems, which suggests that maybe redox functionality is necessary to be over-enhanced in large biochemical systems because you need more energy to fuel them. Or at least that's one hypothesis about why that structure looks the way it is. And also that life evolves to harness more and more energy sources over time because, those are the environments to expand into. So I think there's a lot of arguments for why we see redox having the behavior it does. And we don't know which is sort of the main explanation, but I think there's a lot of things to try looking at. Great. I like that question.
Thank you.
Great, thank you. Next up is Wilson Colley. Wilson says, thank you, Sara. You mentioned the enormity of possible molecular space. Can we rule out big chunks of it based on the range of possible, for example, planetary conditions, or are most of these combinations still possible in this range of physical conditions?
I think probably it is realistic to say we can rule out large chunks of it. But I still think it's so huge that ruling out large chunks doesn't help you much. And I mean, if you just think about like schnapps, like the main elements in life, and you try to build up combinatorial, the number of molecules, just using those as a function of like molecular weight. I mean, it's just exponentially growing, right? So, people might make arguments that this molecule is not thermodynamically possible, or there's no kinetic route to get to this molecule, but I don't think that really rules out much of the space. And then you also have to think about the fact that part of what biology and technology is doing is finding new routes for accessing spaces that were previously inaccessible. So I always like to make the argument that molecules are technosignatures too, because there are some molecules that are produced on planet Earth today that are only possible because of technology. And so some examples of those are like pharmaceutical drugs like Taxol and things that, the complicated molecular structure is things that, would never be produced abiotically or biologically, but they actually require certain sets of controlled and steered conditions that require intelligence to produce them. So I think it depends on what you're saying is like, where is the impossibility space? Is it impossible abiotically, biologically, or technologically? And there's different, and then there's the impossible physically, which I think is where you're asking the question, but I don't know how much of the space that actually rules out.
Okay, thank you. Next up is a question from Vicky, who I think I saw pop her camera on. Yep, there she is. Go ahead, Vicky.
Hey, yeah, no, I was just asking about the assembly theory and the complexity issue. And my question was, presumably you're at the mercy of preservation in that particular case, right? Because complexity, the sworn enemy of complexity is preservation. So I'm just wondering, is there some way you can kind of back that out and kind of put the molecule together and say, there's enough pieces here that I think this was that?
We're trying to do this to bridge to you, Vicky, I promise. But so what I'd like to be able to do is take you only have some slice of like the simple molecule chemistry or like the atmospheric spectra, and you can actually build tools based on the network structure and assembly to actually infer what other higher complexity molecules or structures might be there. So we're actually trying to work on building those kind of inference tools, but it's a hard inference problem, but I think it is possible. A lot of just because of these sort of system level constraints you get in these reaction networks, you might be able to say that there had to be some higher assembly molecule there. And then also the network itself could be considered an assembly structure. So you can talk about constraints in the network properties. So we were talking about constraints in terms of building up a molecule by bonds, but you can look at a reaction network as an assembly space, and also trajectory data as assembly space. So it's a very broad theory, but it's basically just looking for signatures of information being important.
Okay, great.
So hopefully we'll get there.
Yeah, no, it'd be great because I mean, as you can put DMS up in the atmosphere, and then it probably breaks down into or something simple.
Right.
So yeah, it's a hard question of whether or not you have enough of it preserved to be able to tell that the complexity is there.
Yeah, I don't think you would be able to tell it directly from that molecule having like trace abundance in the atmosphere. I think what you would do is take, if you have really good models of atmospheres and you know enough about the chemistry and you know enough about the spectral data and how to build inference to directly extract network signatures or assembly type features from spectra, then you just be able to try to directly build that inference tool for saying those molecules are there, even though you don't detect them directly. You don't have the signature for that molecule, and you might be able to know it was there.
Cool. Thanks.
I hope. I don't know. We'll see. Working on it. That's good.
All right, great. Thanks. There's a couple more questions still in the chat, but I thought I'd maybe take a break here and just ask if anybody wanted to, who is not typed in the chat, has a question they would like to ask.
And I'll have to go in about 5 minutes because I have to pick up from school.
Oh, yeah.
I'm sorry, I can't stay longer. A hard stop there.
That's an important priority. So yes. All right. So this is your last chance if you have something to ask, Sara. All right. Well, in the interest of time, why don't we go to the next question, which is from Devajyoti. I'm sorry if I'm butchering your name. Sorry about that. But he or she asks, do you think we know all the relevant physical quantities required to determine the physical laws for life? Or we may need to find new ones too?
I think we need new ones. So I think this is part of my emphasis on, what's the observable. I think a lot of people do complex system science for thinking about life in a vacuum, where they're just like, life is properties of computation or complexity, and they're never thinking about the observables. But I think what we're really trying to do with assembly theory is build from the observable properties of systems to try to build the theory. And I think that's an important feature. But what are the observables? In assembly theory, we have a clear idea of the shortest path length because you can observe it exactly in molecules. But it's not exactly observable in a human, I don't think, or, other sorts of more complex structures. But then also all the work that we're doing on these statistical patterns of biochemistry is to build new observables that we can actually try to map the theory to, because, as a theoretical physicist coming in and trying to understand sort of fundamental properties of life as it exists in chemical space, there were no observables to map theory to. So most of my group, even though it's a theory group, has been doing a lot of data science and data analysis to try to build observables that we can actually do from that more foundational perspective. So I think you have to do both at the same time.
All right, great, thanks. And our last question is from Meng Li, who I see has turned on his camera. So
Yeah.
Yes, I will definitely ask by myself because I definitely would like to say that Veronica actually worked in Jody's lab, and so definitely say hi to her. And
I will.
It's very exciting to hear your talk because definitely everyone probably thinks about what life really is. I noticed that in your presentation that most of your analysis actually focused on cell systems where you have a cell membrane in there.
Yeah.
So my question would be, what does the viruses and the cell-free system come in terms of the physical laws that applies to all life? And what information does the lipid bilayer provide? And would life need a physical barrier or compartment for them to do life-specific biochemistry?
Yeah, so I think the answer to your last question is yes, at a certain phase of evolution. So I don't think cells came first. I think they're an evolutionary byproduct. And I think they're important for certain classes of reaction in the same way that humans designed round bottom flasks for doing certain classes of reaction, because you need to contain reactions for that certain things to happen. You need those boundary conditions. So I think that's an invention of biology, not the other way around. But as far as like viruses, for example, you can talk about viruses in assembly theory, just like you can talk about any other molecular structure. So viruses have a high assembly index, therefore they are part of life, right? Or they're biosignatures. And so from these sort of statistical patterns, we have thought about getting viral data and putting it in our scaling laws. We just haven't done it yet, but there's no reason that you couldn't, for viruses that have some metabolic functionality, put the reactions there and see, sort of how they do. And it might be the fact that, having a compartment makes their sort of scaling behavior different than what we would expect based on our sort of naive predictions from having bounded systems. But we haven't looked at that yet. But I think all of these things definitely need to be encompassed in it. So the definition of life the theory of life, the theory that explains life, not a definition, not a theory of life, but a theory that you could derive properties we associate to life from, I think should be sufficiently abstract and deep that it should explain a lot of these features. So in principle, what we're trying to do in my group is map it to as many features as we have students to work on that feature, which is good because we have a large, diverse, intellectually, thriving group, but there's only so much you can cover in a short amount of time. But I think those are good goals.
Can I add a comment to that? So I talked to someone who's very into viruses. Then I noticed that there's one thing virus really lacks is they don't have an independent bioenergetic pathway to sustain their life. I don't know why, but that's probably some question that you and your group in the future can look into.
Yeah, no, I think that's a great point. I think that when we say like a cell is a unit of life, that's also a misnomer, because a cell is a informational system that keeps like building itself again and again and again. So it's basically a lineage of information. And then if you look at like asexually reproducing organisms, it's very clear there's no boundary of an individual because they just, divide cells. So from the framework we're thinking about, the units of life are actually the evolutionary lineages, the lineages of information in matter, not the individuals. And viruses don't have any problems in that framework because they're just a pattern of information basically propagating in the biosphere, just like everything else. But the energetic point there about why they survive, whereas other patterns have to be more directly coupled to their energy source, is kind of interesting. So yeah, cool.
Well, thank you.
Great. Thank you very much for the question. It was great.
All right, we should probably wrap this up here now. So
yeah, thank you all so much.
Yeah, thank you for sticking around so long and answering all the questions. Clearly,
thanks for.
That's great. So anyway, yeah, thanks again for coming. And anybody who wants to show any applause, please feel free to do so. And we'll let you go now. So.